Ximing is incredibly humble. He is the leading geneticist within this field, but anytime you go with him with an idea or a question, takes it into consideration. He provides really constructive criticism, but in a way that makes you feel like appreciated. He really wants you to flourish and thrive. So not only does he dedicate himself to the research that he's been doing, but to his students and to all of his peers as well.
When I arrived here in the lab in 1991, I noticed this area was economically depressed. I remember seeing houses on the main street in Port Norris was boarded up. I later learned that this county used to be one of the richest counties in the U.S. because of the oyster fishery and because of overfishing disease and habitat loss. That industry basically disappeared. That caused a lot of economic hardship to the community here. Learned that the disease still a major problem in Delaware Bay for the oysters in Delaware Bay, and I thought this is a big problem and we need to solve it. Triploid oysters have 3 sets of chromosomes. They are sterile, they grow faster, they have a better meat quantity in the summer, so the oyster farmers like to grow triploid. To make that triploid, we need another special oyster called tetraploid oyster. Those tetraploid have 4 sets of chromosomes. In 1993, Rutgers was the first one to create that special tetraploid oyster. And now triploid oyster that was produced from the Rutgers technology account for over 50% of the oysters cultured worldwide. Oysters are very fascinating organisms to work with. To a lot of people, they looks like a rock, not mobile. But inside the oyster is an amazing organism. The oysters are very resilient. They can survive out of water for weeks, sometimes months if you keep them cool. They can survive under the summer sun. And it's just fascinating to me, how can they do that? We spent years of studying oysters and trying to understand the mechanism why they are so resilient.
His work revolutionized shellfish aquaculture and especially oyster aquaculture. The development of the triploid and tetraploid oyster here really revolutionized what these farmers could do. Having triploid seed available allowed them to get oysters to market size in a much shorter amount of time than their diploid counterparts. And the tetraploid technology that he developed allowed hatcheries to have these tetraploid oysters make their own triploid seed for farmers to utilize as well. So not only has he impacted farmers here in the Delaware Bay tremendously and in a positive manner, but he in the whole Northeast region as well. Dr. Guo will get his hands dirty. And it doesn't matter the location, the situation. There have been times where we've been out on boats in pouring rain up to our knees in mud. And I think that's another thing that's really important, is that instead of sort of isolating yourself to one aspect of the research, he is willing to get involved no matter what level he reaches. And he is at the highest level, yet he'll still do the work that a seasonal will do too. And I think that really shows his character, but also builds up teamwork tremendously. I would say without him, I wouldn't be in the position I am in today.
To see years of your work making a real impact on the industry is rewarding. And once an oyster farmer in Maine told me that she would be out of business without Rutgers disease-resistant oysters. That's really gratifying to hear, since we work on make the impact on the industry. I started my career here on Delaware Bay. It has a special place in my heart. With climate change, oysters facing a lot of challenges. I hope the next generation of scientists can understand oysters better, bring them back to Delaware Bay.